Hi everyone, I'm Sandra and I'm a Year 3 PhD candidate at Young Lulin School of Medicine. I work with HLTRP under the guidance of Prof. Sorrentino. And today I welcome you all to a day in my life as a computational biologist trying to bridge AI supercomputing to experimental biology. People often imagine biochemistry as someone who's preparing all day. That is part of the story, but I'm also someone who tries to save this time, money, and energy, trying to figure out drugs using my computer and then test it out in the lab. Most of my mornings start right here at level 5 of Center for Life Science at my desk. As you can see, my computational screens, they always perform something, even if you just see some codes or some random pictures of proteins. I heavily rely on NSCC, which is the National Supercomputing Facility, or NUS HPC High Performance Cluster, to perform tasks before they actually hit the laboratory. Instead of trying them all out in experiments, in order to save time and money, we first try to test them out experimentally, but on computers. A computer can actually function like a human body, trying to mimic a human body's temperature, volume, pressure, and so on. So why not have a look at them before you actually take it to the laboratory and start testing them out? So to do this, I use certain software, but on top of that, I also develop my own. So this means that I integrate datasets, run simulations, analyze results, and generate predictions more efficiently. One of the aspects I enjoy the most of this research that I do is that I get to wear multiple hats. Some days I'm a scientist, as you all know, but some days I'm a programmer. I had to hit certain nights at 2 a.m., 3 a.m. trying to debug everything because my script decided not to work anymore. But computational predictions are only useful if they actually mean something in real world. So to find that out, after I have made some computational predictions, I come right here, which is level 4 of Center for Life Science, where I actually test them out in cells. Now this I do either by myself or with my friends or my lab mates, aka my collaborators, and they help me find out if these actually work or not. I often describe my research as a feedback loop. So once I have a series of compounds that predict to be working, and then I test them out and I see if they are actually working or not, and say it works. I take them back to my computers, try to modify them a little bit, and that way it can help me boost the IC50, the inhibitory concentration of a drug, to inhibit a protein at 50% of the activity. So in order to boost its performance, I can actually change a little bit of those molecules, like the fragments or the functional groups, etc. So to do that, I need some predictions and then validate them in the lab, and then I take back to edit them. Now, this is a continuous loop, so we can call it a feedback loop, where we actually take back and forth and see which is better. This might involve testing compounds in cells, measuring protein activity, or say NAD levels, evaluating biological responses, or confirming whether a predicted interaction actually occurs experimentally. So the big question is, what's the impact of this research? Like, why am I doing all this? So there are certain proteins or certain amino acid regions that are involved in different reactions concerning enzymes, say concerning proteins, that contribute to aging or age-related diseases, or age-related conditions, for example, proteostasis. So these conditions can be altered using drugs, say natural compounds or drug-like molecules that could eventually be developed into novel molecules that actually can alter these conditions. Now, when you think about this, you can actually play around molecules using a computational model, like using an AI model or using existing software, and finally change them into potent molecules like that can inhibit, modulate, activate these proteins. Now that's what I do. I play around with these molecules a lot and try to screen these molecules against a specific target protein that might be involved in, say, Alzheimer's or, say, psychiatric conditions that contribute to aging, like depression. And that's the exciting part of it, that it can actually have a lot of translational impact. Apart from my PhD, I'm actively involved in HLTRP's YSC, Young Scientists Club Committee, where we actually get to involve with a lot of talented minds from diverse backgrounds, and we all have the same mission, that is to increase health span for the common people, shared goals, and as we age, why not improve the quality of life? One of the main things I appreciate a lot about YSC is the opportunity to interact with scientists, clinicians, industry partners, and students who all bring diverse levels of expertise so that we all collaborate with each other as we grow, but this time we grow more healthy. When I'm not doing my work, I mean not staring at my computer screen or not performing any experiments, I usually find time to hit the gym or fit in some therapy sessions into my calendar. Research can be challenging, but the more you do it, you find it more engaging and fun. And even the smallest of discoveries or inventions that you do today will be contributing to a great part of the scientific community that's advancing day by day. And in science, even small discoveries means a lot. Whether through artificial intelligence or supercomputing or my laboratory expertise, the only goal I want to achieve in my scientific life is to turn these discoveries into inventions that once can help the humankind live a better life, or say age better. If you would like to learn more about me or my research, you can always connect with me via my LinkedIn or contact me through email. And I'll be moderating today's Q&A sessions. You can leave your questions and comments in the chat. I'm looking forward to meeting you all and hearing from you soon. Thank you.